This protocol is an easy way to generate the profiles of ubiquitin and ubiquitin-like post-translational modifications by identifying and semi-quantifying specifically purified proteins. So the main advantage of this technique is the use of lentiviruses to create stable cell line of expression within a week and the use of 2 tags to specifically purified proteins. In fact, this kind of post-translational modifications are involved in most biological functions, and alterations of these mechanisms are found in most diseases. Therefore, identifying these alterations may serve as prognostic and/or diagnosis, and of course, as molecular targets. Ubiquitin is one of the most conserved proteins in eukaryotic cells and is essential for their survival. Therefore, this kind of method could be applied to any eukaryotic model, from mammals to yeast. However, our lentiviral system is restricted to the study of mammalian cells. Since the whole procedure is quite long, with sometimes long incubation time, it is possible to do it over 2 days instead of 1. The eluate from nickel beads can be frozen at -20 and before adding the flak beads. This protocol contains many steps, among which some are critical in order to guarantee the final success of the purification and so the accurate establishment of the post-translational modification profiles. Demonstrating of the procedure will be performed by Mirna, Swayden, and Aurélie Dobric, 2 PhD students of the lab.
To begin, add 0.5 × 10⁶ of HEC293 T cells in a 6-well plate with 2 ml per well of DMEM with 10% FBS to seed.